Hello everyone, hope that you are good. Today I will talk about the replication strategy of coronavirus 2. It's not very different from other RNA viruses, but there are some differences and I will just talk about these differences in a while. Um, if you look at the genome, this is the single-stranded RNA genome of the SARS coronavirus 2, and its size is about 30 kb. And if you look at the size of the virus, it's not a small virus comparatively. It's bigger in size when you compare it with hepatitis C virus or other viruses, which are quite smaller. It's almost 3 times bigger in size than the hepatitis C virus, which is also an RNA virus, and the genome of the hepatitis C virus is also a plus-strand mRNA genome. So here it's a plus-strand mRNA genome. And what is a plus-stranded RNA genome? It's a thin strand of RNA which can be readily translated into different kind of proteins in the cytoplasm. The transcription of genes which is taking place, inside the cell, uh, results in mRNA production or messenger RNA production, which is then converted into different kind of proteins in the cytoplasm. So here, if you look at this genome, these are, from 5' to 3'. The genome is encoding for different non-structural proteins followed by the structural proteins. So from here to here, you are, looking at this picture, you can see different kind of non-structural proteins. Non-structural proteins are the proteins that are not taking part in making the structure of this particular virus, but are involved in some enzymatic activities. They are different kind of enzymes. So you can see these different proteases and replicases, helicases, endoribonucleases, which are encoded by the genome of this particular virus. So initially at the start, you can see from here to this particular place, all these are the non-structural proteins or different enzymes encoded by them. And it's a separate, you can see there are two reading frames with the frame shift. I would talk about them, which are encoding these different kind of enzymes, and these should be translated initially. This is the preparatory of this particular virus to make these proteins initially in order to sustain itself inside the cell. And these are the structural proteins which will be formed later on during its life cycle or its replication. So some of the proteins are important, like this papain-like protease, which has now been discovered to be very much important with respect to, the progression of the disease. Because when this virus enters into some particular cells, it would just, initially, what does it do? That it would just block the antiviral response of that particular cells, and that ensures its, survival inside the cell. And this particular protease is not only involved in the proteolytic activity initially, this protease is also involved in suppression of the antiviral response of the cell, and that's why this virus perpetuates, and it leads to this cytokine storm later on and severe phenotype. So this is a very much important protease. You must remember about it. And here you can see other proteases and replicases. Here you can see this is the spike protein, It's one of the largest protein here, and it has two subunits. One is called S1 and the other is called S2. And these subunits later should be separated from each other because they are activated. S1 is involved in this attachment and penetration, and S2 is also involved later on in its the fusion of the membranes with the cellular membranes in order to release the genome of this particular virus. So this is the structure of the coronavirus 2. If you look at the structure, these are the spike protein S1 and S2 that I was talking about. This is the nucleocapsid. Okay, this is the mRNA complex for these capsid proteins, which is collectively called the nucleocapsid, which is inside this particle. This is the membrane which is surrounding this particular nucleocapsid. Then there is envelope protein again, the periphery here, and there is single-stranded RNA, which is plus sense, which is just like mRNA, which is here inside this particular cell. So if you look at the structure of this particular, protein, the spike proteins, which are making this corona-like structure, and that's why it is called coronavirus. Here, this particular spot, this bluish spot, is the receptor binding domain. This is the receptor angiotensin converting enzyme 2, or ACE2 receptor, which has a binding domain here for this particular peptide, part of the peptide. So this is the binding domain of the spike protein. These are the two subunits. The blue one is the S1, and the greener parts are the S2 domain. So these are separated during this replication process. So what happens that its entry into the cell is just like other viruses, but there is slight difference. But because this RNA binding, this receptor binding domain that we are talking about is not exposed as it is exposed in the case of other viruses, and it's also a survival strategy of this virus that it can evade immune responses as a result of that. And it is this is activated as a result of some host enzymes like furin, which are part of the membrane as well, which can cleave the surface protein into these two subunits that we are talking about. And then S1 is, as it is involved in attachment with these angiotensin ACE2 receptor. So the S1 binding domains will bind to these particular receptor ACE2, and this particle would be internalized. So this endosome now, which would be internalized, would be the surface subunit S2 is involved in the fusion of this outer membrane with the membrane of the cell, the plasma membrane, and that would ensure the RNA, only the RNA to come out into the cytoplasm. So this is the mRNA which has entered the cytoplasm now. So this this is a plus strain mRNA. What happens initially that as it enters into the cytoplasm, there are host ribosomes in the cytoplasm. The ribosomes will attach to these this mRNA, and it would start translation of this particular mRNA. So translation actually is taking place, like in two phases. And in the first phase, where the translation is taking place here, the replicases and translation of viral peptides to different reading frames, polypeptide 1 and polypeptide 1AB, with a frame shift are made initially. So what is happening here? Enzyme, replicase, which is required for replication of this particular mRNA later in its life cycle, and other polyproteins, polyprotein 1A and polyprotein 1AB, would be translated here. And these peptides, when these peptides are formed, as the proteases are also translated initially here. So the proteases would cleave the polypeptides. The two polypeptides that are made would be cleaved by the proteases that are translated as a result of translation of this mRNA. And these polypeptides, which are which would cleave, the proteases which would cleave the polypeptide would result in the formation of these different proteins. These, proteins are required for replication and transcription, are enzymes required for replication and transcription complex. Okay. So non-structural protein 3, 4, and 6 are formed as a result of proteolysis of this particular polypeptide, which is made here. Now all the enzymes are available. RNA, the plus-strand mRNA is also available. What happens to the plus-strand mRNA next is this plus-strand mRNA would have two fates, either this plus-strand mRNA would be replicated to make a negative sense RNA, a copy of the negative sense RNA, or this plus-strand mRNA would be, utilized by this replication transcription complex of enzymes to make these genomic and subgenomic RNAs, So the subgenomic RNAs, what are the subgenomic RNAs? As a result of transcription, if this mRNA is transcribed, these different mRNA molecules of different sizes will be made there, and these are coding for different proteins, like the structural proteins, nucleic acid, and the spike protein, the membrane protein, envelope protein. These would be transcribed out of this RNA. This is called the subgenomic RNA transcription. It's a discontinuous type of transcription that I would talk about. And uh, uh, as I told you, this genomic RNA, the negative sense RNA, which is made out of this plus-strand RNA, can either be transcribed into making these subgenomic RNAs, or the plus-strand RNA, or it can be replicated to make the plus-strand RNA. Okay, so remember these two steps. Don't be confused with it. First, the RNA, RCT, or the replication transcription complex, converted this plus-strand mRNA into a negative sense RNA, and this negative sense RNA is transcribed by the replication transcription complex into to either make the plus-strand RNA again, just like this one, or it is transcribed, it is replicated to make the plus-strand RNA, or it is transcribed to make, these different kind of subgenomic RNAs, which are making the structural proteins. So these subgenomic RNAs, when these are formed, as these are making different structural proteins, the structural proteins would be, formed here in the endoplasmic reticulum. You can see these transcription of viral structural proteins, S, M, and E, etc. This thing would happen in the endoplasmic reticulum. Okay, and then the maturation of these proteins would take place here, and their assembly would take place here in structures which are known as ERGC, or the endoplasmic reticulum Golgi intermediate complexes, where these proteins and this mRNA, which has been synthesized here, would be packaged in the form of a full-fledged, virion. This is the virion which has now to be exocytosed, or to be taken out of the cell, okay, because this is mature now. So what's happening? It's entering its membrane fuse, endocytosis take place, mRNA just is coming here into the cytoplasm, and first it is transcribed into polyproteins, translated into polyproteins. The polyproteins are cleaved to make these different non-structural proteins. So these have different enzymes, and the these enzyme complex are the replication or replicase and transcription responsible enzymes complex, which is made here, would convert it into a negative sense RNA molecule, which again would be converted by the same complex into either a plus strand RNA, which is to be packaged in this particle, or into subgenomic RNAs, which are giving rise to these different kind of proteins, in the endoplasmic reticulum, and then they mature here and make a particle, a full-fledged virion, is these different proteins are assembled in a fashion or in a way to enclose this RNA genome, and this infectious particle would just go out of the cell. So this is the mechanism that this particular virus is used. And here, uh, you know, uh, this particular, just like all other, viruses, it is making a use of the host machinery or the polymerase here to carry out this particular job, or the host ribosomes here initially. And again, unlike some of the viruses like hepatitis B virus, it's not getting into the nucleus. This entire scene is happening inside the cytoplasm, and it is just like it happens in the case of hepatitis C virus and other viruses, that most of them are utilizing this strategy. Their replication and their transcription, etc., it's taking place in the cytoplasm, inside these bodies, endoplasmic reticulum, and the Golgi complexes. And then, sorry, these particles are just, taken out of the cell. So this is a bit a detail of, this process, that this is the plus strand RNA, which has to be, translated into polyprotein 1A and polyprotein 1AB. So these are 1A and 1B polyproteins. And then proteolysis of these two bigger proteins take place to make these smaller proteins that you see here. These smaller proteins are different replicases, transcriptase proteins, is you have seen the genome of the virus is encoding for it. So these polyproteins are bigger in size. These are cleaved into smaller parts, which are performing different functions. They are enzymes doing replication or transcription. So these enzymes then make these complexes, which are known as replication transcription complexes. And these complexes then join, they would attach to this plus strand mRNA. And this plus strand mRNA is converted into a negative sense mRNA here. And this negative sense mRNA either would be packaged into this new virion, which is formed in the ERGC, or this would be, transcribed into making these different mRNAs, which are coding for different structural proteins, because the structural protein are required for making the structure of this particle. So these structural proteins would, surround this plus strand mRNA to make an infectious particle. As I told you initially, that it is a discontinuous transcription. So how this thing is happening, these mRNA molecules of different sizes are made here. So if you look at it, this is the entire genome, 5' to 3' here are the leader sequences. Okay, this is the open reading frame 1, and then there is a frame shift, ribosomal frame shift site here, and then there is open reading frame 1b. So one protein polypeptide which is formed is 1a, and other polypeptide is 1a plus 1b, 1ab. Okay, some part of 1a and b. So these are the non-structural proteins coding for all these enzymes which are initially required by the virus to sustain inside the cell, or to do in other activities like replication and transcription. So these are first of all, these polypeptides are made, and these polypeptides are then uh, cleaved by different proteases, as you see here. Proteolysis of them take place into making these different enzymes, which are carrying the rest of activity, as you see. And these are the structural proteins. The later part towards the 3' of this particular virus are coding for these different structural proteins: A, 2B, 3, 4, 5, 6, 7, etc. Okay. So these are all the proteins that are making the structure of this particular particle. Okay. So what is a discontinuous transcription? Suppose this is an mRNA molecule, and at the start here, the leader sequence, there is a leader TRS, or transcription regulatory signal. And for making these structural proteins, here you can see these are body TRSs, or body transcription regulatory signals. So the transcription, what is happening around here when this transcription start here, uh, you know, uh, this process starts here. The enzyme moves from this place to this particular place, and then it jumps from here to here. So this part, the leader sequence, would be part of this particular proteins. For instance, this is protein number 7, which is formed here. This is the mRNA, this the smallest mRNA that you see here, how it has been transcribed. So it has been transcribed like this. When the transcription starts and enzyme moves from here to this particular TRS, transcription regulatory signal, the enzyme just jumps from here to here. So this would become initial part of this particular mRNA to make this particular protein number 7. Now, when the number 6 or number 5 mRNA is to be synthesized, this is coding for another non-structural proteins. The enzyme would start from here, and if it is number 5, the enzyme would just move to the 7, 6 up to this signal, the TR is number 5, and then the enzyme would jump to this particular part to make it part of it, to transcribe it or make it part of protein number 5. So this part plus this part would become this particular mRNA, and you can see here it will be translated into a different kind of protein. So this is called a discontinuous transcription, which is happening here in the case of this particular virus. So it was all about the replication of this particular coronavirus 2. And as I told you, it's slightly different from other viruses. And due to this discontinuous transcription strategy, but it is happening in the cytoplasm by utilizing different, host factors, host ribosomes, and some enzymes initially, and then, making its own enzymes and carrying on its process of replication and transcription to make different proteins and packaging. All these things are happening in the endoplasmic reticulum and Golgi complexes. So that's all for today from my side. If you have any question regarding this particular lecture, so please let me know about it. I would be very happy to answer them. Thank you so much.